here with this great excitement. I think that we've had this as one of our strategic goals, is to truly embed translational research into our trial activities. And so we've been working on it over a number of years, and really this is now our official launch. So I'm really pleased to introduce both Anna De Fazio as the chair of the TR-ANZGOG committee and Claire Davies as the project manager, who will be able to run through the TR-ANZGOG and what will mean for all of us as part of the ANZGOG trials group. So I think I'm handing over to Claire first, or Anna.
Yes, to Anna, to me. Can you hear me?
We can.
Thank you. So thank you very much for the opportunity to speak today. So Link, can we move to the first slide? Thank you. So it gives me really so much pleasure to speak to you today to launch TR-ANZGOG and to acknowledge all of the hard work that's gone into developing this really important new ANZGOG initiative. Before we go through the slides, I'd like to make a few opening remarks to set the scene and to put TR-ANZGOG into context. So when TR-ANZGOG was first conceived, not that many years ago, the translational aspects of most clinical trials were an optional extra. Sites had the option to the extent to which they could participate, ticking box A, B, and C, depending on their capacity. So our original thoughts about TR-ANZGOG were to provide the mechanisms to allow all sites to be able to fully participate in the translational arms of all ANZGOG trials. So we anticipated at that time that the importance of translational aims of clinical trials would increase, and it was clear that ANZGOG needed to be prepared for what was coming. And in fact, that's really what we've seen. Biospecimens for molecular testing and the translational arms have really become front and center of many of our trials, and most have at least some aspect of biospecimen collection and have translational aims. So the first and really very important aim of TR-ANZGOG is to support investigators to achieve the translational aims of their clinical trial. Secondly, we need to think ahead. We need to think about what will happen with the samples and data once the trial is over. Every single clinical trial is a huge investment, from the women who so generously consent to participate in the trial, to the investigators, the ANZGOG and OASIS teams, the funding bodies, drug companies, regulatory bodies. Look, the list is absolutely endless. And with such a huge investment in both time, money, and commitment, we really have a responsibility to ensure that we make the most of all of the information that can be obtained from every single trial. And that includes from the samples that are collected. There are potential reasons in a clinical trial, for instance, why one patient might respond incredibly well to a treatment or intervention, while another is resistant. And that might not be known at the time that the trial is being conducted and might surface into the future. The samples from the trial would be incredibly valuable in addressing these questions, but there are often significant barriers to using clinical trial specimens in research. For example, the wording of the consent form, for instance, it may be as simple as that, may not be quite right to allow future studies. Or the samples might be locked away in the freezer of the basement of the institution where the principal investigator was at the time of the trial. And that investigator has since moved on to the next trial. So really, TR-ANZGOG aims to provide enduring custodianship for biospecimens and to maximise the use of biospecimens through research. Can I have the next slide, please? So we've had a large amount of consultation with the sector, and Claire will go through this later, but I'll just briefly describe what has come from that in terms of the overarching themes of TR-ANZGOG. So first and foremost, our main focus is to improve outcomes for women with gynaecological cancer. Sometimes we can be caught up in a lot of the issues and the barriers and the regulations, but I think, you know, if we understand at the forefront that that is what our overall aim is, I think it helps to deal with many of those day-to-day issues. So we want to implement processes that will enable translational research and remove those barriers, be inclusive, transparent, and have good governance. We want to make sure that there's consistency in quality because of course that will ultimately make sure that the quality of the research outcomes is very high. We want to be able to promote those large question-driven collaborative and translational research projects. And underpinning all of that is to take a very ethical approach with respect for privacy and consideration for equity and cultural values. Next slide, please. So we've developed a series of principles. So our not quite 10 commandments, only 8. But I'll quickly go through these because, again, our guiding principles help us to ensure that we can achieve our aims. So in terms of clinical trial support, one of our principles is that access to trial support will be via a transparent and equitable application process. Samples will be embargoed until the translational aspects of the specific trial are complete. And of course, each trial will be a little bit different. And so we'll ensure to make sure that that any differences in the translational aspects from one trial to another are really taken into account. So not, you know, like everything, it won't be one size fits all. We really want to do whatever we can to maximise the translational outcomes for each trial. Next slide, please. In terms of biospecimen access, we really want to make sure that we've got all of the approvals and mechanisms in place right from the get-go. It's been very difficult. It's very difficult to retrofit processes to be able to use samples that were collected in the past. So we really want to fix that and ensure that all of the appropriate consents and approvals are obtained up front, so that that the patients understand and are happy for what's being proposed, and that's all very clear, so that any barriers that might come up are taken care of very early in the piece. Certainly there will be, one of the issues is that often any research that it's outside of the scope of the original trial is often not allowed, but we're hoping to be able to do that, to manage it through the TR-ANZGOG Trial and Support Access Committee, because there's a lot of issues to consider. And we need, one of our really big principles is that it's a principle of collaboration, not duplication in TR-ANZGOG, because the samples are very finite. So we need to balance being able to learn as much as we can from these finite, valuable resources, but we want them to be used, not locked away. So really the principles of collaboration, not duplication, but a willingness for the samples to actually get out there and be used. And we've looked at some guidance, or we're providing some guidance around what would be sort of the sweet spot, if you like, in terms of what samples can be obtained and what are the most useful for future translational research. But of course, that will be very different again from trial to trial. Next slide, please. And of course, in any of these things, there's a huge amount of input that we value and recognise from at all points along the pathway. And so we recognise at the get-go that we will acknowledge the significant time and intellectual input at all points of the pathway, and recognise the costs. We know that we can't, the individual sites can't do this with no funding, and for it to really work properly, we have to recognise that that these sorts of initiatives come at a cost. So we need to provide a solid financial base, make sure that the translational aspects are built into grant applications to underpin funding, obtain some separate grant funding if that's necessary, and of course to ensure that acknowledgement of everyone in publications and in other fora. So these are our guiding principles. Next slide, please. So how are we going to do all that? So there are, you will have seen many of these slides before, 4 aspects of TR-ANZGOG. The first is the resource portal. So the resource portal is a repository for all of our methods, operating procedures, so that people can pick these up to put into their different trials without everyone having to reinvent the wheel. Document templates, patient information consent forms, advice for grant and trial budgets, because it's always difficult to ensure that the translational aspects are budgeted appropriately, an inventory of our specimens, and application forms for each of the different aspects. So that portal, I'd like to, I'm very happy to announce is now live. So many of those documents are now available on the website, and you'll be all sent a password to be able to go into those site to access those documents, either the steering committee or all of the ANZGOG members. Next slide, please. As I've mentioned, one of the really important aspects of TR-ANZGOG is to really support clinical trialists in being able to undertake their translational work. And of course, this doesn't mean that TR-ANZGOG will be able to do everything. Really, it's we'll be collaborating with CTC and trial PIs to work out what all of the best mechanisms will be and what help will be needed and what things will be undertaken by others. So our essential, but I've mentioned a couple of times, really, we're going to be providing advice on wording to put into consent documents, etc., so that these samples can be used into the future. Next slide, please. One of the aspects, the very first aspect I mentioned, was that support for different sites to be able to participate fully in translational research. And sometimes that's a matter of having people in laboratories, in biobanks that are co-located on some of the sites to be able to help to collect those specimens. So we have created a network of TR-ANZGOG laboratories that will be able to assist in those features. Next slide, please. And of course, the translational researchers. So we've established mechanisms to be able to apply to TR-ANZGOG for the use of biospecimens and research data into the future. We've looked at data management. So we're getting essential data sets to go along with each of the biospecimens. And these have been aligned with a number of very successful studies that have been done in the past, AOCS, Innovate, Otter, Solus 2. So taking into consideration many of the studies that have gone on in the past to really, again, make sure that we have that essential data set confirmed that needs to go along with the biospecimens, because of course that is really critical. Another point that really needs to be considered up front are that the data sharing needs to be in those very, very initial collaborative agreements. So we've done quite a lot of work and are continuing to do work in this aspect to make sure that that is all done appropriately. Next slide, please. So, of course, I really hardly have to tell this group about what the potential benefits of translational outcomes are through something like TR-ANZGOG. I think we're all very well aware of those, but really just to reiterate that the TR-ANZGOG initiative has the potential to improve outcomes for women with gynaecological cancer in 3 key ways: to understand the clinical and molecular drivers that underpin response to current and future therapies, to collaboratively investigate novel scientific questions that need to be addressed in future clinical trials, and really to promote those big questions that can only be addressed by large collaborative translational studies. Next slide, please. So with that, I'd like to hand over to Claire Davies. I have to say none of this could have been possible without the enormous amount of work that Claire has done and the fantastic support that she's received from Alison Evans and from everyone at ANZGOG. So I'll just hand over now to Claire to tell you about a little bit more about how we've gotten to this point. Thank you.
Thank you, Anna. I'm conscious of everyone's time as well with the RAC meeting starting after this. So I'll be brief, but I just wanted to really take this opportunity to provide a little overview of the process and the milestones to reach this point in time and the next steps going forward. Next slide. So TR-ANZGOG started with a discovery phase, which, as Phil mentioned, was some years back, but 2016 when Anna first proposed this concept, and there was a workshop that formed where draft aims and principles were established and presented at the 2017 ASM. And I have to give a big thanks to Dr. Helen Gooden for her work in preparing a position paper, which really assessed the feasibility and the interest and the need within the sector for TR-ANZGOG. We were fortunate to secure bequest seed funding. I started in August 2018, shortly followed by our first steering committee meeting. Then we received more philanthropic funding at the end of 2018, and we were also fortunate to be awarded a grant through OHMR for the New South Wales Biobank provision of services. And that coincided with us registering and commencing a certification program that's internationally recognised through one of the Canadian groups, OBA and CTRNet. And then there was another further steering committee meeting at the start of 2019 before we started further broader sector consultation. Next slide. So this extended sector consultation over the past couple of years, I've talked to different groups. Huge thanks to everyone for their input and expertise, time. It's really been a broad group of collaborators who have contributed to us reaching this point in TR-ANZGOG. And I just want to bring everyone's attention to the TR-ANZGOG consensus workshop that was held at the end of last year. Next slide. So this consensus workshop was attended by 40 experts representing clinical, scientific, legal, ethical, consumer and operational disciplines. And this consumer workshop really had the key aims to create a robust foundation for TR-ANZGOG and its activities, achieve clarity and consensus on the key governance and process issues, and agree the engagement and communication needed to support TR-ANZGOG and empower the TR-ANZGOG community. So we had focus areas that were discussed on the day in the areas of custodianship and the governance of data and specimens, specimen collection, data management, laboratory network, and consent guidelines. And a particularly valuable outcome from this workshop was the formation of 3 working groups in the areas of patient consent, the labs, and the data management, who have really assisted with the development, the review, and the approval of key documents. Next slide. So as building on this development phase of documents, SOLACE 2 was used as a pilot study, which really helped test out some of the processes. This phase 2 clinical trial, which looks at immune priming in recurrent ovarian cancer and chaired by Professor Claire Scott, has a heavy translational focus for which TR-ANZGOG was managing this component, which involves 114 patients being recruited over 15 sites with 26 time points for the translational bloods, as well as tissue and ascites. So that's really helped develop some of these key resources and processes for TR-ANZGOG going forward. Next slide. So I'm happy to say that, as Anna stated and Phil mentioned, that these documents are now completed. So these key documents are available on the TR-ANZGOG webpage of the ANZGOG website. These documents include background documents such as the principles and the governance and our terms of reference, and also the approval process for how our laboratory network is going to be established, and also resource documents for use for investigators, for translational researchers. And so you'll see all of those listed on the website. And as I mentioned, I will be shortly sending around a password to enable the member-only access to these portals. Next slide. So the next steps going forward, really, as we start implementing TR-ANZGOG and integrating with trials currently in development. So I'll be working with our collaborating centres and our trial PIs. And I say I, but that's under the stewardship of the whole of the TR-ANZGOG Steering Committee. But I look forward to working with everyone to really integrate TR-ANZGOG so that we can future-proof the value of these specimens and the data collected and maximise the investment that's being made. We'll also be seeking standalone ethics approval independent of the trial, so that will really assure participants that the correct processes and policies are in place. As I mentioned, with the TR-ANZGOG network of laboratories, establishment of this has already commenced, and we are currently undergoing the application and approval process. These laboratories will then have a collaborative research agreement in place with ANZGOG, And so then we will have a light touch, trial-specific agreement for each trial dependent on the trial's requirements and the lab's capacity at that point in time. And further to this, we'll have more vendor negotiations, which includes procurement of our laboratory information management systems to really have optimal management of sample tracking. And I'm also looking into doing vendor negotiations for different consumables and resources to help with budgeting going forward. Next slide. I just wanted to leave you with this concluding quote from Anna, which really resonated with me when I commenced. Clinical trials tell us whether a particular drug or intervention improves survival. TR-ANZGOG will help us to investigate why the drug tested in the trial works or doesn't work at an individual patient level. For me, that really just highlights how TR-ANZGOG will complement the clinical trial process. Next slide. And huge thanks and acknowledgement to everyone who has helped to reach this milestone, this exciting point in time, particularly members of the working groups. You've really contributed your expertise and your time to help with these policies and processes. And of course to Alison Evans and Anna, who have been massive help with guidance and stewardship of this research initiative. Thank you very much. Next slide. So thank you. I'm conscious that the RAC will start in about 5 minutes. So perhaps if anyone has any questions, I guess if there's maybe time for one now, but otherwise, please feel free to direct those to Anna and I, and we'll be able to liaise with you directly offline.
Okay, well, thank you very much, Claire and Anna, for an incredible effort to date. Indeed, outlining that history, I think, does tell us, as you say, both the time and energy that's gone into this over the last 3 to 4 years, and particularly over the last 2 years. So really grateful to both of you for all of that effort, and really it's fantastic we've reached this point now. So thank you very much, and we'll look forward to next year and how this moves forward to the benefit of both the trial activity and indeed to research more generally. So yes, you're right, we're up against it in terms of time. We do have a RAC meeting starting in 5 minutes. So really, I think it is just my final duty to thank you all for attending. It is not straightforward doing this by Zoom, because there really is difficult to have some of the intimate questionings that we might like to have, and the opportunity for members. But I think we certainly would invite anyone who's got comments or questions, particularly with the TR-ANZGOG, to come back to Claire or Anna or anyone on there within ANZGOG, and indeed any of the presentations throughout the AGM. So thank you all. There is a short break, and then there is the RAC meeting starting at 2. Once again, I just need to highlight these dates that are here. This was up at the beginning of the meeting, but for some of those who might have missed it, I think in particular we have the education series, the preceptorship, and indeed the ASM next year, which will be a virtual meeting. It'll be held over 2 days, as is listed there. Friday the 12th of February is the main conference program, which will be based on the program from this year. And that is moving forward, but we have a second day, which is the Pure Science Symposium as listed there. So please make a note of those now, and the early bird registration is until the 21st of December.